Ahead here. So I think everyone should be trickling in. I'd just like to welcome everyone who's joining us. We'll just give it another minute or so, just because I opened up the webinar. I want to ensure that everyone is here. So we'll start in just 1 minute. Okay, I think that's good. The number of attendees joining has stopped, so I suspect we'll have some more joining us soon. But I would like to get started. And first, I'd like to welcome everyone for joining our 6th session of the BioCanRx Summer Speaker Series. This was a series that was developed in lieu of our face-to-face conference, our conference Summit for Cancer Immunotherapy that was to take place in November 2020. And instead of that, we have done this series to give an opportunity for our funded network investigators to present their research. And it's a great opportunity to have our network and many different stakeholders and participants attend these sessions. So we're switching up a little bit in this session. And today we are having Dr. John Bell and Dr. Jennifer Queasy present on something a little bit different. So they'll be presenting Fighting Viruses with Viruses, Repurposing Cancer Vaccine Platforms to Fight the Spread of COVID-19. Before we jump into things, I would like to introduce our next Summit Speaker Series, and this will be the 7th session. And this will be by Dr. Ali Khan Lalani at McMaster University, and he'll be presenting on his cytoshrink clinical trial. So we don't have the information posted to our website just yet, but do stay tuned for more information. And in a few moments, you'll be receiving this information regarding our website in the chat box. So just a few housekeeping notes for today. I would just like to let you know that the session is being recorded and it will be available on our website after the fact. And you can also find all of our previously recorded sessions there as well. On the website, you can also find the speaker bios and more information about today's session. And with respect to the technical aspects for today, if you hover over the bottom, you'll be able to see the chat functionalities. And I'd ask that if you have any questions, you ask them in the Q&A box, and this will provide an opportunity for other participants to upvote questions that they have of interest. So in the case that we run out of time for questions, I'll prioritize those that have the most upvotes. If you would prefer to ask your questions orally, then please raise your hand and I'll unmute you, and that gives you an opportunity to ask your questions to John and Jen orally. And so with that, I would like to introduce our 2 speakers for today. The first speaker will be Dr. John Bell, and he is BioCanRx's scientific director. He's a scientist at the OHRI, and he's also a professor at the University of Ottawa. And we also have Dr. Jennifer Queasy, who is the director of biomanufacturing operations with BioCanRx, and she's the director of the Biotherapeutics Manufacturing Center at the Ottawa Hospital Research Institute, and in particular with the virus manufacturing facility. So with that, John, I'd like to hand it over to you to take it from there. And I'll stop sharing.
Okay, thanks, Megan. Happy to talk to everyone today. Let me see if I can make sure this all works. All right. Okay, that's the right orientation, Megan?
Yep, looks good.
Okay, so really what I'm going to talk about today is some work that we've done to pivot from making cancer therapeutics to COVID vaccines. And the context here is that Megan sort of said to me one day, you know, what on earth are you guys doing making infectious disease vaccines when you're cancer biologists? So I thought, okay, I'll explain to her what our thinking was. But really, I think what's happened in the world is we were all headed in one direction, and then suddenly COVID happened. And so when that happened, we decided as a group, a number of us, to get together and try to see if we couldn't do something to convert some of our technology that we had been using in the cancer space to create a vaccine for COVID. And just to mention that this work was supported by CIHR through a grant we were very fortunate to get from CIHR, but also from a FAST grant that we got that was in fact supported by the Thistledown Foundation, which is Shopify group in Ottawa, who put money into the FAST grant program. So really appreciate the support that they gave us. So anyhow, the bottom line is, you know, why would you think that you should be involved in making a COVID vaccine? And before let me see, make sure I'm doing this right. I just want, whoops, go backwards here. I just want to mention that this is really actually a tremendous team effort. A lot of different people, and they're not all on the slide actually, were involved in the grant application that we put in and have participated in helping to do it. And I think what happened is we were sort of somewhat out of the lab, couldn't work for a little while. And at the same time, we all work on cancer therapeutics and recognize that, you know, cancer patients are probably some of the most susceptible people to having a very bad outcome from a COVID infection. So we really wanted to try to do something to repurpose some of what we were doing. And I just want to acknowledge my co-applicants on this CHR grant with Doug Mahoney in Calgary, Ray-Jean Lapointe in Montreal, Carolina Ilkow, and my colleagues here in Ottawa. And this picture of Ray-Jean is actually quite interesting because he looks very distinguished there, but I noticed when we talk every week on the phone that he's getting a very good COVID haircut. So it's down to about here now. And so that's not what he really looks like. But anyhow, all I have to say, a great group of people that have been working together and I think have made some fantastic progress. So as you know, there are a lot of different kinds of technologies out there being developed for a COVID vaccine. Things like live attenuated viruses, which basically it would be the SARS-CoV-2 virus itself, but somehow inactivated or attenuated in some way. Non-replicating viral vectors. Replicating viral vectors are being tested by a number of groups, and this is just taken from Nature back in the fall, and you can see there's a lot of activity happening at that time. Since that time, of course, as we all know, there's now 4 that are approved in Canada for use. 2 of these are RNA-based vaccines from Pfizer and Moderna, and 2 are non-replicating viruses from AstraZeneca and from Oxford and from J&J, which is using a chimp ad, and J&J, which is using another version of adenovirus 26. I think what's interesting about this is it's great news. I mean, we're all really excited this is going to happen and we're going to actually get vaccinated. What we don't know is, are these vaccines going to be good enough in the long term? That's an open question still. Are they going to be able to deal with some of the variants that are rising in the population? But what you've also seen, I think, which is quite interesting, is the challenges with getting any biotherapeutic to the clinic, and that is manufacturing is an issue. It's been an issue for all these various platforms. I mean, you might have seen recently that J&J is talking about the fact that they're having some manufacturing issues. So I think it's timely to have this discussion today about the sort of challenges associated with doing manufacturing and what we're doing. One of the things that I want to speak about today is that the platforms we're developing, I think, are a little easier to manufacture than the ones that are currently available for a variety of reasons, and also just the way that they're basically based upon vectors which have been used for many, many years as vaccines, and so we know a lot more about them. So why do we need so many vaccine strategies? And I think it's you know, one obvious thing is that it's a new virus that has not really been impacting the human population before, so we don't really know exactly how best to deal with it. Although after the first SARS virus, there have been a number of vaccines that were developed, although never actually taken through to commercialization because governments felt it was not worth their investment to do something which they thought would never come back, and unfortunately it's come back in spades now with SARS-CoV-2. I think it's also unclear even still today, although I'm very, very optimistic that the RNA vaccines are going to be fine and the attenuated viruses will be good, but it's unclear at this point if that's going to be the best approach. Are variants going to arise that are going to be able to escape the antibodies or the immune response that's created by the current vaccines? We don't really understand the immunopathology of SARS-CoV-2 as well as we need to yet, because there's you know, things that we see in animal models and things that we see in people, and we're not really sure exactly what's going on. Normally, this is what we think about when we're making a vaccine, is there's a virus like SARS-CoV-2, and we generate neutralizing antibodies through a vaccine strategy, and those neutralizing antibodies will either kill this virus particle or, for instance, keep it from binding to its cellular receptor. But we know that in the case of some viruses, and possibly SARS-CoV-2, that if you don't generate the right kind of antibody, if you make a non-neutralizing antibody, they may actually facilitate virus entry into target cells, and that may actually make things worse. So perhaps an anemic immune response may be worse than having no immune response, because you might get this antibody-dependent enhancement of virus replication or cause a massive cytokine pathology. So it really is important, I think, for, even though we have some vaccines out there, it's really important for the world to continue to work on this and make sure that when we're ready for the next pandemic, or we're ready if this one should reoccur because of variants that arise. We also think that it's maybe not, what we're currently seeing is the RNA vaccines or double dose vaccines, which are one after the other. J&J argues that theirs is a single dose vaccine, and I think Oxford thinks to some extent it could be as well. But, do we need to do some sort of boosting situation? Should we use a homologous boost where you give the same vaccine multiple times, or a heterologous boost? Because we know that a heterologous boost with two different vectors often generates much better and more potent immune responses. And so maybe that's going to be required for some kinds of people who are maybe older people or cancer patients who need to have an extra zip in their immune response. So what are the challenges to creating an effective vaccine for SARS-CoV-2 and actually for cancer? And I think that's the sort of perspective we came at it from already, is cancer has some overlaps with SARS-CoV-2 and the challenges to making a vaccine. And so we thought some of the know-how that we've done and approaches we've developed would be useful for this approach. So first of all, you really need to generate a rapid and hopefully sterilizing immune response. And that's true for both SARS-CoV-2 and, well, maybe not the rapid part, but the sterilizing response for both SARS-CoV-2 and cancer, because in cancer, you don't want the tumor to respond to come back again. You really want to eliminate all of it. So that's something that you really probably need to do for both these indications. We know that virus variants are already rising during the pandemic because we don't have it under control. And we also know that cancer cells evolve. So you want to make sure that you have a vaccine that's going to be able to deal with that particular issue. And so one way to do that is to make a very potent vaccine, but also maybe one that has multiple targets that it hits, so that even if you have a variant arise, same as with a cancer mutating, that you have an immune response that can deal with that kind of variation. The route of administration is something we're interested in, because where do you want that immune response? So in the cancer situation, you want it in the tumor microenvironment. Maybe in a SARS-CoV situation, you really want to have a really robust immune response in your respiratory tract, because that's the mode of entry into the individual. Safety is the same for both of them. You have to have something that's very, very safe, although some might argue that for something that's going to be given to a healthy individual, you even have to raise that bar a little bit higher, potentially, although we try to make things that we think are very safe, independent of whether or not you're a cancer patient or not, but clearly that's an important issue. The ease to manufacture has become obvious to the world now that manufacturing can be a challenge, and one has to really take that into account when creating a vaccine. And the other thing you now hear about is cold chain, which is this issue where you have to be able to transport the vaccine to sites, and you have to store it, for instance, -80. You have to reformulate it and get it ready to inject. How long is it going to be stable for? This is also true of cancer therapeutics, but maybe not to the same extent, because cancer centers often have a -80 freezers, and so maybe it's not as big an issue. But for both kinds, I think it's an important thing to think about. So our thinking behind the vaccine design, we wanted to go with something that was different. These would be replicating viruses, which we have already been working on as cancer therapeutic viruses. They're replicating, but they're highly attenuated, so they don't really have the capacity to spread within normal tissues very well at all. They just really, once they enter a normal cell, they have a very short replication cycle that is then aborted. And so we thought that viruses like this that encode COVID antigens may be very effective at inducing potent immune responses, because the actual act of having at least an abortive replication is a really strong signal within the body to respond immunologically. So there is a rationale for using these kinds of viruses, we believe. Using a heterologous prime boost strategy might be needed to generate really potent immune responses. We don't know yet what's going to be required, and there may be certain, as I mentioned, certain populations that need something more than just a single or double homologous boost. You may need to have something more substantial for older patients, maybe, or for, as I mentioned, people with compromised immune systems. I think what really we believe that's very important is that you have to have both an antibody and a T cell response, and it should be against multiple targets. Because as we've seen already, variants will arise in a pandemic. If you're only targeting one protein, like the S protein, which all 4 vaccines do, maybe that won't be enough, because maybe mutations in the S protein will allow it to escape, for instance, antibody neutralization. So really, we want to make sure that we get something that does both those things to help us deal with the possibility of virus variant escape. We think that replicating viruses, because they have some capacity to replicate at the site of injection, it may be possible to deliver very low doses, and that may be really important in trying to reduce manufacturing concerns. So just to speak to this concept of heterologous prime boost and the relationship between cancer therapy and infectious disease immunotherapy, this is a slide which actually is used. I think my colleague Brian Lichty may have created this one. I can't remember who made it, but this was really a technology that was developed several years ago for cancer therapy from our collaboration with a great group at Master. It was Brian and Byron Bridle at the time and Young Han Wan. And the concept was that you'd prime an immune response with a particular virus backbone. In this case, it was an adenovirus expressing a tumor antigen. And then you'd boost it with a second virus backbone, completely immunologically distinct from the first one, but encoding the same tumor antigen. And the idea was that you'd focus the immune response in the boost on the tumor antigen and not on the vectors. And of course, in the cancer therapies, we were also overlaying that with the fact that the second virus was replication competent and could be an oncolytic. And this is just some data here. Actually, I'm just cite the guys down here who were involved in this work that we were developing at that time, looking at a B16 melanoma model. And this is a model which really is refractory to therapy by the viruses we're using as individual agents. But when we used them in this context of using a heterologous prime and boost, what we had again was a vaccine vector which encoded a tumor antigen. This is topochromatopteromerase or DCT. And then we would follow that up with an IV administration of an oncolytic virus that also encoded the same antigen. And so if you just primed with adenovirus, we saw sort of a good response to the tumor antigen, but not tremendous. This is just looking at the percentage of interferon expressing CD8 T cells against DCT. But when we used this heterologous, and if we used adenovirus followed by adenovirus, this really didn't change at all. But when we used an adenovirus followed by the Maraba virus, we saw that we had tremendous immune responses in these animals, up towards the 40 to 50% of their T cells were actually directed against this, really, which was essentially a self-antigen. So this is sort of the thing that was in our mind as we were thinking about trying to make a COVID therapeutic, is that we knew that this kind of heterologous prime boost with two different kinds of vaccines might be very effective at generating really tremendous immune responses, and that may be important. Now, we are generating, as I'll talk about in a minute, multiple vaccine candidates, but we can imagine that these could also be used with combination with the current vaccines that are already approved. So what virus vectors did we use? We used things that we actually had been using and developing clinically as cancer therapeutics. This particular actually was rhabdovirus family. The one I just showed you on the other slide was Maraba virus, which is a version of a rhabdovirus. This is another one, VSV, vesicular stomatitis virus. And we liked it as a vaccine vector because it's been used. It is, as you probably all know, this was developed in Canada by the group in Winnipeg. Heinz Feldman was involved in that. Dr. Plummer was involved in developing this at one point. And this is really has become an FDA-approved replicating vaccine for Ebola virus. It was the one that was the most effective. It's small. It's only about 12 kb. It's a single-strand RNA genome. It's negative sense, which means it has to be transcribed before it can be translated. Really only has 5 genes. It does have some coding capacity, though. You can put about 4 kb of a transgene into that. It replicates very rapidly, and I think one of the things we always loved about this as a cancer therapeutic as well as a vaccine is it's very easy to manufacture, and it's a cytoplasmic virus, so no chance of neurotoxicity, genotoxicity. So that was one of our candidates. The other candidate was a vaccinia virus platform. This is TN10, which is actually a virus that was used as the smallpox vaccine in China for millions of people. So it has had a long track record in China. It's been used many times to generate all kinds of different kinds of therapeutics, whether they're oncolytics or viruses expressing things like Ebola virus, GP to make vaccines. And so we thought it had some good legs in it that would make sense for us. As a double-stranded DNA virus has quite large and has a huge coding capacity. So one can imagine doing lots of different things with that genetically. And it was a virus platform that we've been working in the lab to make it an oncolytic virus therapeutic. So I'm going to speak first about this rhabdovirus work that was done actually in Doug Mahoney's lab in Calgary, and Kyle Potts was a very bright young postdoc in Doug's lab who's really led the charge on this. And so this is VSV. It normally looks like this. It's a bullet-shaped particle. It has a glycoprotein on its surface, which many of you have heard about, which allows it to infect cells. This is the SARS-CoV-2 virus, which has a more complex genome, but just of note, it has something on the surface called spike, which is analogous to the G protein on VSV, as well as other proteins, which we'll talk about later, N, M, and E, and so on. But this spike protein is involved in mediating the binding of this virus to human cells and leading to infection in SARS-CoV-2. So what Doug did was create a chimeric virus, which no longer had G on its surface. It completely eliminated G and replaced it with spike. So he's now making a spike expressing virus, which is dependent upon binding to human cells through the spike protein of SARS-CoV-2. And so the dream here was that this virus would replicate, it would be recognized as a foreign antigen, and it would generate an immune response against the S protein. And I'm not going to show you all the data because it's some lovely data that Doug and Kyle have got, but I really want them to have the chance to present. But just as a sort of a synopsis of what they found is that this virus is really good. It's very safe, even if it's given intranasally in hamsters and macaques so far. But really, as we had predicted, very low doses provide extremely robust neutralizing titers. And this happens, a single dose gives very long-lasting responses. And as we'd hoped, it is a very easy to manufacture kind of product. So there's a lot of biology behind this that I think Doug will talk about at another time, but really exciting because this is a one-step potential vaccine. The single dose could be very effective. Now, Merck was generating something very similar to this. And they abandoned it because they found when they gave this thing intramuscularly, it was unable to vaccinate because it couldn't really replicate in the muscle because the muscle didn't have the ACE2 receptor. So we've been really championing this as an intranasal administrated virus as a vaccine because the nasal passages do have the receptor for it. And so far, things do look pretty promising. I just want to also mention to anybody who's interested in virus neutralization assays, which are critical for following vaccine activity. The Mahoney group has also automated this now, and they have a really nice high throughput way of doing it. You should contact Doug at Calgary if you're interested at all, but this is really a great system he's got set up to rapidly analyze patient responses or vaccine responses in animal models. The other thing we thought about is what targets are we going to use in a vaccine, and this again is work by Ray Jean, who's in our project. And Ray Jean is a T-cell biologist, mostly interested in cancer aspects of T-cell biology, but he got involved with us in doing things like LE spots to look at what are the epitopes that are responded to in any sort of immune setting. And so we know that when SARS-CoV-2 infects, it expresses a number of different kinds of protein targets. So we just were focusing on the S, which is the spike protein, but also the M, N, E, and other non-structural proteins that are found in the virion. And what Rejan's been doing is looking at convalescent patients from Montreal who have responded and have recovered from an infection of SARS-CoV-2 to see which kinds of epitopes are actually being recognized by patients. And the thinking is those are the things that we should incorporate into our vaccines because those are the appropriate targets. And again, I'm not going to show Rejan's data because he'll want to show that one later, but what he's in the summary shown is that he can see that patients respond to S to a lower degree actually, and much more robustly to N and M, two of the structural proteins in the virus. So that really tells us if we're going to make a vaccine, we probably should incorporate all three of these proteins to some extent if we want to make the most broadly active vaccine. And Regen's got a beautiful system for mapping the exact epitopes and regions that are recognized, and it's allowing us to design viruses or vaccines that will respond to generate both potent antibody responses against S, but also potent T cell responses. Lastly, I want to speak about the vaccinia virus, TN10, that we are developing as a vaccine. And this is work which really been driven by Carolina Ilkow, a colleague of mine here at the OHRI. Vaccinia is a really interesting virus. We are using it as an oncolytic as well. It has a very interesting life cycle, which I really won't have time to talk about today, but just to say that it's a really interesting cytoplasmic virus that has a lot of capacity to be used to express a variety of antigens. And in fact, it has been used as a smallpox vaccine, but many other kinds of vaccines as well. One of the things that we're really interested in is can you use the great coding capacity of vaccinia virus to create other kinds of vaccine vectors inside the body? And so one work, this is sort of project that was being developed in my lab for a cancer application. In this case, a self-amplifying RNA is encoded at some place within the vaccinia virus genome, and at a distant site, structural proteins, which would make a virion component that would encapsulate that RNA polymerase, that self-amplifying RNA, are then put together in the same infected cell. And what comes out is a virus-like particle that also has a self-amplifying RNA in it to really generate potent immune responses or to transfer other kinds of therapeutic genes in the cancer setting. And this is work done by Nicholas Martin. And what he's shown here is in this vaccinia virus, which encodes a self-amplifying RNA, which expresses RFP, that when he starts an infection on for instance, a spheroid, you see that the outside begins to be infected and express vaccinia proteins, but very rapidly, these VLPs are also expressed from this infected cell, and ultimately you get tremendous expression of the VLP within the tumor spheroid. And so our thinking is, can we also do this to create vaccines in situ by using a very attenuated vaccinia virus that in essence makes a SARS-like VLP particle in situ and generate potent immune responses. And so that's ongoing work which we're pretty excited about. But just to sort of end with a little bit of data in this short period of time we have to talk, just to say that we've made a number of different kinds of vaccines, and they all look to be very, very safe in animal models that we've looked in macaques, in mice and hamsters. So they all have very good safety profile, and including even direct injection into the brain in young mice using the vaccinia virus. So it looks very encouraging from that perspective. And back to this concept of heterologous prime boost, where we could use these two vectors in tandem to try to generate the most potent possible immune response. And this is sort of just taken from textbooks that shows you that when you do a primary and then a boost, if you use two different vectors, you often get a much more potent immune response. And so this might be important for COVID-19 because maybe to get sterilized immunization, we need to really ramp up the immune response. Maybe, as we mentioned, suboptimal antibody responses can be an issue. And so we may have to combine our two virus vaccines together or combine them with other vaccines to get the best possible outcomes. Well, that remains to be seen. But just to show you that we've got some really nice immune responses. So what we're looking at here are neutralizing antibodies. So the way this assay is done is we take a virus like the one that Doug generated, which is VSV with a spike on its surface. We immunize animals and prime them and then boost them. And then we look to see what kind of immune response they generate against the S protein. And in this particular neutralization assay, we use 2,000 plaque-forming units of virus. We mix it with dilutions of serum from vaccinated animals or from patients, and we look to see what sort of immune response we get. And what's interesting is with the vaccinia virus, we actually generate pretty good inhibition. Essentially looks like really able to inhibit 100% of the 2,000 virus particles we put in there, even when we dilute the serum 500-fold, up to 2,000-fold at 50% inhibition, which is pretty strong, I think, just from a single injection. If we use homologous prime boost, where we give both the vaccines twice, both intranasally, we see even better immune responses. But the best is probably when we use the combination of both an intranasal prime with vaccinia and a VSV boost intranasally. And we see really getting tremendous immune responses where the serum can be diluted out 2,000-fold and still is able to neutralize all of the virus in the assay. So I don't have enough time to show you lots of other data which is coming out, but it looks like some of our ideas were probably correct, and that it is possible to use this approach to generate really potent immune responses. And we have a couple different virus vectors which we think will be useful. And now why virus vectors, just to sort of segue to what Jen's going to talk about, the one thing that's nice about these is they're very easy to manufacture. These viruses, because they're replicating, they grow to high titers, they're easy to purify. And because they're replicating, we believe we're going to be able to use much lower doses to vaccinate with. So that's really what we were thinking about from the beginning. And as you'll now hear from Jennifer, we wanted to be able to be in a position where if we had a vaccine that was good, we could rapidly manufacture it. And fortunately, we've been manufacturing cancer-based vaccines for some time now. And so we're able to quickly transition to this kind of vaccine technology to what we have available in Ottawa. So Jen, do you want to take it away here?
Great, thanks so much, John. I am just going to start sharing my screen. I just want to confirm with BioKinetics.
Looks good, Jen.
Okay, great. Thanks so much, Megan. Okay, great. So like John was mentioning, they're working hard on the innovation side of things in the lab, number of collaborations that, has been involved in getting the candidates that they're working on right now to the stage that they're at. But before we can obviously put that innovation into a person, there's a whole other area of research that needs to happen, and that's the process development research, which really provides us with the step-by-step process of how we actually manufacture this under GMP conditions to a scale and a purity that's required in order to safely administer that innovation into people. And so whether that's the one or the both, of the two candidates that John's talking about, regardless, this is where the Biotherapeutics Manufacturing Center comes in. So specifically the virus manufacturing arm of that. But really quickly, what is biomanufacturing? Biomanufacturing uses biologic systems to manufacture or produce whatever the product is. So within the health field, we use cells to manufacture therapeutic viruses. How does this differ from pharmaceuticals? Biologic therapies are typically a lot more complex to manufacture. They require oftentimes larger facilities, more specialized equipment, and are very highly reliant on very highly trained personnel. Now, it's not to say that for pharmaceuticals, that's not the case. You don't need highly trained personnel, you absolutely do. But it's to a different level because, again, the order of magnitude of complexity is that much more. So in the case of pharmaceutical processes, these are typically chemical processes, synthetics, versus these highly complex and living organism sort of systems that we use with biomanufacturing. And so we look to biomanufacturing as offering the bench to bottle solution. So obviously this stops short of the bedside, because of course there's that extra layer of translation that has to happen. But really for biomanufacturing, we're using these biologic systems to manufacture a number of different types of novel therapies. Now, I'd be remiss if I didn't acknowledge, obviously, that we're living in a very different world today than we were even a year ago. And so biomanufacturing is obviously critically important to manufacturing the billions of doses of vaccine that are going to have to be manufactured for some semblance of normalcy to return to the world. But I think we'd also be remiss if we look to the COVID-19 vaccine efforts as being, the entire picture when it came to biomanufacturing, because it is but the tip of this iceberg. Biomanufacturing enables the production of cell-based, virus-based, antibody-based therapeutics that extend way outside of infectious diseases to other indications, such as cancer, regenerative medicine applications, and rare disease. So pre-COVID, with the advent of biologic medicines, biomanufacturing had unprecedented growth. The demand increased exponentially, and there became a global shortage for access to this sort of capacity. Now, of course, this has been further compounded by the fact that we have to manufacture, again, billions of doses of vaccine. But notwithstanding, biomanufacturing really is that tenuous sort of link between the folks on the research side of things. You see this guy here, and then of course bringing it over to the clinic. And it very much is this rickety looking bridge. It exists. However, it's it's not something that is super stable because the demand is so high that what that does is it increases the cost. And oftentimes this increased cost results in access to this sort of, or to this very important activity not being available for people like researchers, academic researchers, or even the Canadian startup culture. And so in effect, we have this valley of death where the products go to die because we can't manufacture to the scale and purity that's required to take it from a bench research innovation to something that's suitable to go into people. And it's really for this reason, actually, that the VMF exists today. John was showing in some of his slides, reference to the Miraba product that was, an innovation, collaboration between a number of groups. But really, the problem that we encountered with the Miraba is that we could not find an appropriate place to manufacture to the scale and level of purity that's required to put that into a phase 1/2 clinical trial. And so this is where, it was decided that there was a facility at the Ottawa Hospital, that it was easier and less expensive to commission that facility to manufacture that virus than it would be to go externally. And I say easier in parentheses because it was anything but easy, but it was a team of dedicated staff that really did make that happen. And so really the virus manufacturing facility, we see it as this mechanism through which we can accelerate the development of these novel therapies through manufacturing, and of course the focus on training. Now, what we hope that this does, or what we hope this enables, is that it provides access to Canadian academics so they can translate their innovations as well as the innovations of the Canadian startup culture. And of course, it's with the ultimate goal of providing these sorts of novel therapies to the benefit of our ultimate stakeholder, which are the patients. So who is the virus manufacturing facility? So I was using BMC and the VMF interchangeably there. The virus manufacturing facility is actually falls within the Biotherapeutics Manufacturing Center at the Ottawa Hospital Research Institute. And there are two arms, the virus side and the cell manufacturing side. And so because we're talking about vaccine manufacturing or virus-based vaccine manufacturing, and I represent the virus manufacturing group, that is what I will be focusing on today. So the virus manufacturing facility at the Ottawa Hospital has been manufacturing clinical grade therapeutic viruses for the better part of 15 years. For the majority of those 15 years, what we've manufactured has been with an eye to cancer as an indication. And we've been manufacturing in a single GMP suite, which I'm very excited to say in lockstep with the increasing demand, both for different therapeutic indications, but of course, because of COVID, we are expanding that capacity to include a second facility. So it will enable us to manufacture two different virus-based products in parallel, so doubling our capacity on the production side of things. So as I mentioned, we've been manufacturing therapeutic viruses for the last 15 years, and these viruses have subsequently been used in clinical trials, both domestically and abroad. And that includes phase 1, 2, and approval for use in phase 3. And again, this is more within the cancer indication. And so for that reason, we have, once we manufactured for that clinical trial for the Miraba that John was showing, we quickly reoriented to recognize, the need that we had was not unique in the space, that this is something that other academics likewise and other people in the sector were looking for this sort of expertise and capacity. And so we reoriented this facility to become a contract development and manufacturing organization. And our partners, as I mentioned, are academics and they're industry. And within industry, we have startup companies all the way up to large pharma companies. And so as a CDMO, we offer translational facilities and expertise. Our 35 staff have wide breadth of expertise. We have redundant and overlapping expertise, of course, as well. And this all enables us to offer the entire spectrum of manufacturing services that somebody with an innovation such as John's coming in would look for. And so that includes assay development and qualification, process development services, GMP manufacturing, so the actual production, and QC analytical testing and stability studies. To expand our ability to support our partners, we've invested over the years in both adherent and in suspension technologies. So the majority of our work for the last 10 years, 10 to 12 years has been in adherent culture. So we have our roller cell 40, which you can see in the middle here, which is how we manufacture our vaccinia and our rhabdoviruses. And then also we've invested in the iCELLis 500, which is also an adherent-based technology. But what's really exciting about that is it offers about 40 times the surface area of our RC40. So in essence, 40 times the capacity of the RC40. And recently, we've added to our fleet suspension-based technologies. So here's a picture of the STR 200, I call it. It's actually the 500. So this has a 500 liter capacity. What we've onboarded is a 200 liter capacity. All right, so in the case of John, here he is with his COVID vaccine in the flask, and he's coming to the BMC. What would we offer him? So as I mentioned, we have a number of services we can offer, you know, subset or the entire spectrum. And so for John, we would look to initiate this technology transfer with him and his group to understand the virus or viruses, whether it's the vaccinia or the VSV or both. And we would look to adapt that to a process that would be amenable to manufacturing at the scale and purity required for him to use it in clinical testing. We'd also be using the time to develop assays specific to that virus, which would then be qualified for use as a release test in our QC analytics lab. And of course, we would be looking to slot him in and that product into our GMP production suite so that we could manufacture it. Now, just to go back to process development, oftentimes process development is a lot like this figure. It's organized chaos. There are a multitude of parameters that you can test to optimize growth conditions, as well as downstream purification conditions. But what we offer at the BMC, our competitive advantage is the fact that we have been manufacturing these oncolytic viruses for the last 15 years, and a number of different types of oncolytic viruses, so like the rhabdoviruses and vaccinia that John was speaking about. And so what we do is we leverage existing processes specific to the virus that comes to us, and we make small optimizations or alterations based on the characteristics of that specific virus, maybe because of the addition of transgenes or the removal of other things. And so just to show you, we've extensive experience manufacturing rhabdoviruses and pox viruses, and just showing you here on the right of the screen, just basically an idea, of one of the processes that we use using these RC40s, and just with an eye to, we're able to accelerate the development of these sorts of products again because we leverage these existing processes that we have an extensive amount of experience working with. So I think that where I'm going to leave it off is talking about the importance of the GMP team that we built. Because we are a GMP facility, really our quality is built into the product at every stage. And so every single one of the staff, whether they are in production or process development, they are involved in building the quality into our products. It can take years to properly train a GMP operator. And it's because very much the training has to be dynamic and it has to evolve. One, because you subscribe to current good manufacturing practices, and that's a commitment to continuous improvement, but also to make sure that we're a nimble facility and we're able to service the needs of our partners. We're looking to continuously onboard new technologies to support the manufacturing of a number of different vaccines and oncolytic viruses and other types of products. And so in lockstep with that, we had recognized that the best way to ensure that we have the right people at the right time was to build a pipeline of highly qualified personnel. And so we've done that in a first of its kind training program where we bring on interns for an 8-month paid position where we will train them for the 8 months in the entire spectrum of the manufacturing activities that we do at the BMC. We've been running this program successfully for the last 2 years. And over those 2 years, we've had 10 trainees join us. And I'm very excited to say that as our organization has grown, has created the opportunities for us to keep all 10 of those highly qualified personnel. And we have just completed our most recent cycle of hiring for these interns and are excited to welcome 5 additional staff joining us in this program starting in May. So perhaps I'll leave it at that because I think we're getting close to time, and I know that we wanted to leave some time for some questions.
Great, thank you, Jen. John, did you want to turn your camera back on and we can start into the Q&A period of the session? Awesome. So John, I'll start with you. There were a few questions that came in as you were talking, and I'll try to do them in order that they came in. And I do note that there is one that will be asked orally as well, but the questions in the Q&A are, the first one is from David Latulippe, and it's, when you say easy to manufacture, does this mean in terms of both upstream and downstream, or just upstream?
Yeah, no, it's a good question, and when I say easy to manufacture, speaking relatively speaking, to making other things, there are challenges with manufacturing viruses for sure, which David's well aware of, and one of the challenges is actually the purification step downstream. So we've worked hard over the years to get the highest possible yield we can from cells that are infected. But then when you go downstream, it becomes a bit of a challenge because often in some viruses which are cell associated, you have to separate the cell debris and the DNA from the virus product. And so that requires lots of different kinds of cleaning steps, including column chromatography, which we're constantly working to optimize. But I think it's actually because we've created such a good upstream production yield, of the virus, it makes the downstream a little bit easier because the doses for vaccine are much lower, so it makes it possible to purify it. But in fact, there are certainly challenges to purifying viruses that are really important to consider as well.
Thanks. Next question from Alice in Danville is, do you know the relative levels of the T cell responses to the S, N, and P proteins? Are responses to N and P as frequent as those to S, or is S the immune dominant protein?
So this is Rajan's work, which I don't want to go into in detail because I think it's, we haven't published it yet, and I want him to have the opportunity. But just to say that it surprised us that in fact, although S is obviously the main target for the antibody response, in convalescent patients we saw in Montreal, it was not the main response for T cells. And in fact, N and M were much more important in that T cell response. So it does say to us, at least from this early look, that you really want to have a vaccine that probably is more than just S. Although clearly there are T cell responses against S, it would look like N and M, which are less likely to be variants, were maybe better targets.
We'll have to have Réjean come on and present his data. Okay, and now moving, it's in the chat, but it was the next question asked from Scott. So when you are combining replicative viruses with elements of SARS-CoV-2, how do you ensure you don't inadvertently create a harmful or contagious virus?
I didn't quite get the first part, Megan.
Sorry, I mumbled a little bit. When you are combining replicative viruses with elements of SARS-CoV-2, how do you ensure you don't inadvertently create a harmful or contagious virus?
No, it's a great question. It's a very good question, because whenever you're starting to make chimeric viruses, you could be creating something that could be incredibly dangerous. And so in this particular case, what we do is we use a backbone, which is extremely attenuated. And it'd be very challenging for that virus backbone on its own to revert to become more of a wild type virus. And so we think that by overlaying some of these proteins, which are not particularly involved in, are not necessarily increasing the virulence of the protein, simply the targeting of it, that it shouldn't be an issue. But that's certainly something that we're very aware of and are very careful about looking at. But a great example would be Ebola virus on VSV, where in fact, Ebola virus puts terror in everyone's mind when they think about it. And the Ebola virus G protein on VSV was perfectly safe.
Correct. Going back to chat, a question from Chelsea Jackson is, if viral vectored vaccines become a widely applied technology, would you expect to see a decreased efficiency due to multiple vaccines, therapeutics, potentially using the same vector? And the question is, especially if the prime and boost are two distinct vectors.
Great question, and that, of course, that's always been a limitation to using virus-based vaccines, is you may get a situation where you start to generate immune responses against the vectors, which make them less useful. Although I will note that several people are, for instance, making measles-based COVID vaccines, so there is still a possibility to use these. My dream would be that the RNA vaccines work really well, and we never have to worry about this again. And so that would be the very best case scenario in many respects, if we can get RNA-based vaccines that really are potent and generate good T-cell responses as well as antibody responses. I think that's the way of the future. We just aren't there yet. We're doing that experiment in real time, basically. So I hope that maybe we can abandon this particular approach for infectious disease. But in the meantime, rather want to keep on top of this in case it turns out that RNA vaccines are not all that we dreamed they would be, and we do have to have a different platform.
Okay, great. I think one last one in the chat and then a few others. So, and just a reminder, folks, if you do want to have your questions upvoted, because we're nearing the end of question period, do write them in the Q&A. So a question from Julian Zmansky is, are the vaccinia spike expressing VLPs able to infect cells via ACE2?
We haven't formally tested that yet, Julian. That's happening in the next little while for sure. But we do know that other VLPs that we make in this way do have the appropriate targeting to the appropriate receptor. So we would expect that would be the case, but we haven't formally shown that yet.
Perfect. Okay, and maybe two more questions for you, John, and then I've got a couple of questions for Jen as well. And then I do have a question here that I think is a great end from Peter. So, so with respect to you, John, is there a limit to how many vaccines an individual can receive over a lifetime? I.e., is there a limit to immunological memory?
That's a great question. I don't know the answer to that honestly, but I suspect that it can go on quite well, except as you start to get older, then probably you have diminished capacity. I think that's well known. But I don't think it's a matter of, if you have 10, you're done. I think that you can have many vaccines, because clearly we get infections all the time, which are sort of natural vaccines, and we continue to respond to them. The exact answer I couldn't give you, but maybe there's somebody else on this call today who's more of an immunologist could actually speak to that. But I suspect we're not going to run out of runway just yet.
Great. And I mean, I do welcome, if there is anyone else on the call that would like to answer, we can certainly make this an engaging, interactive type event. So the person who did have their hand raised doesn't appear to. If you'd like to ask your question, just raise your hand again and we can ask it orally. But I see also a couple of questions coming in the chat function. So again, for John, how does replicative... Yeah, no, I have not asked this. How does replicative capacity differ in recombinant virus, for example, VSV producing S protein? How do we maximize replicating capacity of VSV viruses?
Well, the VSV virus that Doug's created with the S on the surface actually replicates pretty well, almost as good as wild type VSV. It is down a little bit. But it's a good question, do you start to attenuate the viruses, and so you can't manufacture them as well as you want? And one of my colleagues here in Ottawa, Jean-Simon Diallo, has done some tremendous work identifying small molecules that enhance virus production in things like manufacturing cell lines. And so even if you create a virus which is attenuated because of the way you've changed its glycoprotein, it may be possible to still boost manufacturing by using some of these small molecules that JS has created, which he's shown already can boost lentivirus production, AAV production, oncolytic virus production. So I think that would be a workaround if you actually start to cripple the virus too much.
Perfect. Great. So thanks, John. I'm going to turn to Jen now. There are a few questions with respect to manufacturing. So how does the CMO produce commercial scale product, or are you limited to clinical scale? And basically, can you describe that? And do your clients look to larger organizations for clinical scale, i.e. is technology transfer a priority for you?
Great question. Thanks so much. So our niche in manufacturing scale is really to service the early phase clinical trial. And that's, I think, in lockstep with the size of our facility. And it's really what we have grown our expertise on the past 15 years. And so because of that, we have had the benefit of working with partners that have been looking for, phase 3 material. As I mentioned, we have manufactured material that has been approved for use in phase 3, but typically you're looking for a lot larger scale than what we currently have the capacity for at the BMC. And so that's where we would prioritize the technology transfer to commercial CDMOs. We have done that in the past successfully. And so absolutely, that's part of our model is we're looking to enable this at all stages, not just the early phase.
Perfect. Question from Scott McComb, and I apologize, that was from Ian Goff that asked that question. And from Scott, is there any concern in the bioproduction industry about a bubble in viral production capacity coming online in response to the pandemic?
That's a great question. I would say, given the fact that we're still seeing that there's unmet need, no. I think what will be key to ensuring that we don't hit that bubble is that, you are building robust and adaptable processes that maybe aren't just, one virus specific process. Obviously, it's a concern, but I think that in Canada, especially, we're a bit behind other developed nations when it comes to our biologics manufacturing capacity. And so at least within Canada, I think we definitely have a ways to go before we hit that.
Perfect. And we do have a couple of more questions. While I have everyone online, because I know people start to take off around this time, I just wanted to thank you both for presenting. We do have 2 more questions, so don't go anywhere. And a reminder to all of our participants to please fill out the survey at the end of the session. It'll pop up on your screen. And as always, we use those feedback to help inform our subsequent sessions and the format and things like that. So thank you guys for that. So Jen, just back to you with respect to the next question. And actually, this is for both of you. So apologies, I'm just looking for it. So 30,000 foot view question is: Canada is scrambling to recreate adequate domestic vaccine production, as you just talked about. What impact could your approach and facility in Ottawa have on this challenge? And this is from Peter Greer.
Yeah, again, great question. Thanks so much, Peter. So what's interesting is that, as I mentioned, for the last 15 years, we've been manufacturing therapeutic viruses, specifically oncolytic viruses that have been administered in various clinical trials for cancer patients. Now, the therapeutic dose for an oncolytic virus is orders of magnitude higher than what you'd be looking at for a vaccine. Circling back to something that John had mentioned earlier. So we're looking, in the E6, E7 range for a vaccine, whereas for oncolytic viruses, we're talking about, E11. And so we have gotten quite good at manufacturing these replicating attenuated viruses, such that we could make enough in one run. So our 40-liter capacity in our RC40, that we could dose patients with an oncolytic virus. We could dose, I think, upwards of 1,000 patients or more with the 1E11. And so if you translate that back to number of vaccine doses, I would say adapting our existing processes that, in a given run then for a vaccine, we could look to generate hundreds of thousands, if not millions of doses in one run. And I mean, that's if we're dosing at the E6, E7 range. I was speaking with John a little bit earlier today, and that was the range that they were using the VSV EBOV or Ebola vaccine at. And subsequent studies actually showed that you could generate the same sort of immune response if you dose people at like 1E3. So again, just shows you, based on what we've been able to manufacture for oncolytic viruses, that, applying that process, we could generate a very large number of vaccines, even with the modest footprint that we have right now. Again, independent of any sort of expansion, but in our existing capacity.
Maybe I could just add something to that too, Megan, because I think it's a key question right now, and in the time when Canada is thinking about, potentially boosting biomanufacturing. For instance, one model would be a massive facility, but clearly I think what works well in Canada is to have lots of different sites that are working together in a network and providing and being nimble. We don't know if the next pandemic is coming next July. I hope not, but or it's going to come in 50 years, and so we have to have a situation sustainable but flexible. And I think that's what Canada's good at, making, making nimble, responsive, a network of manufacturing facilities. That would make a lot more sense to me than making a, one large facility or centered in one city. I think this is pretty critical. In the United States, there's at least 1,200 GMP facilities making FDA approved material. So, if you just do the math, divided by 10, we should have 120. I'm not saying we should have 120, but we should have a lot, I think, because we need to spread it out to remove the chances of we need redundancy. We need to avoid the chances that any one city is hit hard by a particular pandemic and so on. So I think Peter's identified the right question right now is what's the best way to do it? And I would argue good facilities like ours and others around Canada that have focused on this would be putting them together in a network makes a lot of sense.
Yeah, great last question, I think, to ask on in this current climate. So we are at 3 o'clock sharp. I want to thank you guys both again for participating, and we did get a few thank yous in the chat box as well. So with that being said, Thanks, everyone. Thanks for the participants for attending and for the questions, great questions. Just as a reminder, this will be posted on our website and you can tune in at another time, and our next our next session will also be on our website. So with that, thanks again, and we'll see you next time. Bye both.